I'm Geert-Jan Boons. I'm a professor at the Complex Carbohydrate Research Center of the University of Georgia. And this afternoon I'm going to tell you about a project that focuses on the development of an automated platform for the enzymatic synthesis of complex oligosaccharides. It's performed in collaboration with my colleague Kelly Morman, who's also a professor at the Complex Carbohydrate Research Center, and the project is performed by doctors T-Hai Lee and Lin Liu. The synthesis of complex carbohydrates by enzymes has taken an enormous flight over the last couple of years, and it is now possible to make glycans depicted in this cartoon.
Shown is a complex branched glycan structure with different sugars being represented by different colored shapes.
And this one, for example, was isolated from breast cancer and has the potential to serve as an early marker for breast cancer diagnosis. To develop such diagnostic tools, we need well-defined glycans, and enzymatic synthesis makes this possible. So why is this possible? First of all, the enzymes that make complex carbohydrates in nature, Glycosyl transferases, they are highly regio and stereoselective and can be performed at very mild and easy conditions. Over the last few years, we have identified many microbial and mammalian-derived glycosyl transferases, and these can be easily produced through recombinant expression, and now we have a toolbox that makes it possible to make almost every mammalian glycosyl transferase. Once upon a time, sugar nucleotide donors that are being employed by these enzymes were difficult to obtain. Now we have convenient approaches for enzyme-based production of these reagents. Also, over the last couple of years, universal acceptors have been developed that make it possible to make glycans enzymatically with highly complex branched architectures. So now the time is right to automate glycosyl transferase mediated oligosaccharide synthesis. The paradigm in automation is a solid support to which a growing acceptor is attached. And purification is easily accomplished by filtration and washing. However, we discovered that this approach does not work for enzyme-mediated synthesis. On a solid support, a resin, reactions are much slower because most are actually inside the particle. The enzyme has to migrate into that, and as a result, sugar nucleotide donors are not easily transferred, and instead they are hydrolyzed. To address this issue, we developed a catch and release system. So in a catch and release approach of synthesis, the reactions are performed in solution, and purification is accomplished by attaching the reagent through an affinity tag to a solid phase extraction resin, and then by washing, all excess reagents can be removed and the next step can be performed. This sounds easy, but it is actually very challenging. First of all, we need to identify a tag that can easily be introduced and removed without affecting the complex carbohydrate. We don't want to do any evaporation steps because that would make automation complex. We want a single set of conditions for capture and release, whether it's a monosaccharide or a 20-mer. These conditions should be the same. And finally, we need to develop a platform that makes it possible to automate all these steps. To accomplish these goals, we developed a tag that is based on sulfonates. They contain these negatively charged residues. And we found that such a tag can easily be captured on a commercial diethylamino ethyl resin. So it can be captured, and then by washing with water and a low concentration of ammonium carbonate, all reagents, including excess sugar nucleotide, can be washed away. Then the oligosaccharide can be released from the resin by using a higher concentration of ammonium carbonate, 300 millimolar. And then by adjusting the pH, by just simply adding acetic acid, a buffer is obtained that is appropriate for the next enzymatic reaction. And we have found by adding the right component, that almost every glycosyl transferase works in such a buffer. So the idea is this molecule is being loaded on the resin, we perform washing steps, the molecule of interest, our glycan, stays, and then using this high concentration ammonium carbonate, it comes out, we adjust the pH, and then it can go to the next reaction step. So all of this we perform repetitively on a robot. And we used as a robot a commercial liquid handling system that we rebuilt and added components to make this all work. So the workstation is from ChemSpeed. And it is composed of a temperature controlled rack where the reactions take place, reagents that are also stored in a controlled manner. The solid phase extraction resins are put in another rack, and then we have components for further reagent addition. All is handled by a syringe pump that is that drives a robotic arm that has also a pH needle. So how does this work? We dissolve our substrate, the first monosaccharide, in an ammonium carbonate buffer that is adjusted automatically to pH 7. Then all other components, sugar nucleotide, enzyme, BSA, possible metals are added automatically. The reaction is incubated for a fixed period of time. Then a small sample is taken for later analysis. And then it is automatically transferred to a solid phase extraction column, that the diaminoethyl amine, which is washed with water, low concentration ammonium carbonate to remove all contaminants, and then high concentration of ammonium carbonate to release it. Then it is automatically transferred to the reaction rack for the next reaction. And here you see how the equipment actually looks like. So here you have the various racks for reagents and solid phase extraction columns. Here you see the syringe pump that basically drives a needle that picks up and delivers the reagents wherever they have to go.
On the left side of this slide is shown a chemical reaction to build more complex sugars. Different sugars are represented by different colored shapes. Shown on the right hand of the slide are different retention time showing the peaks of elution for different sugars. As the sugar becomes longer, the retention time is increased and the peak moves from the left to the right.
So here you see an example of how the system works. So we have over here a sugar represented as N-acetyl glucosamine, and then by the addition of an enzyme called B4-GALT1, we can add a first galactose. We perform automatically a solid phase extraction. Then the next enzyme is added, B3GNT2, which adds an N-acetyl glucosamine to install a lactosamine. And this process is now repeated multiple times to synthesize an oligosaccharide. And here you see a trace analysis of each step by HPLC. And you can see that the reactions perform extremely efficiently and that high quality final products are obtained. These compounds can then be further derivatized by using other enzymes, and we can, for example, install a sialic acid at a 2,6 or a 2,3 manner, or we can install an H-type antigen by using a fucosyl transferase. The scope of this methodology is very broad. And as I already mentioned, we have enzymes in place that can make almost every mammalian oligosaccharide.
Shown in this image are various complex sugar structures, both linear and branched, with different sugars in each molecule being represented by different colored shapes.
So we can make poly-lactosamines, we can make human milk oligosaccharides, and again you see these are molecules with highly complex architectures. They are branched, and on each arm there is a unique epitope, and by an orchestrated use of these enzymes we can make these molecules in a highly controlled manner. We can synthesize gangliosides, and these are, for example, implicated in immune suppression, or we can make N-link glycans that are very common components of glycoproteins. The ability to make all these molecules gives really an enormous opportunities to advance glycoscience. We can employ these well-defined glycans to define ligands of glycan-binding proteins. We can use N-link glycans to make glycoforms of glycoproteins, and that is, for example, very important for therapeutic antibody development. We can also use these molecules as analytical standards. It's still very difficult to define exact structures of glycans by analytical methods, and by having these well-defined molecules available, such methods can be further refined. So this is a robust platform that we believe can be implemented in many laboratories. If you want to learn more about this technology, please contact my colleague Kelly Morman or me, and we are more than happy to share reagents such as enzymes or host you in our laboratories to familiarize you with this type of automated synthesis. Thank you for your attention.